Okay, so welcome to our 26th lecture. So today we're going to talk about something new. It's called synthetic biology. So some of you might have seen this term floating around in the internet or in some forum before. So today we're going to explore what synthetic biology is, and it is a design of a synthetic life or biological system. And we're going to look at how this is achieved, like what kind of techniques, what kind of modeling that has to be done to understand such system, and the role of computation in synthetic biology. Okay, so when we talk about synthetic biology, so it's a design of completely, or maybe it's a modification of existing like cells or bacteria. For example, you can change the gene, you can design it so that it can produce maybe the compound that you want, or maybe you want to understand like how the cell operate. We want to understand all the mechanisms inside. You can also start from pieces of DNA, and you want to combine them together so that when you put all these DNA inside an empty cell, you get a working living organism. So the goal of synthetic biology, there are two directions that you can go. The first one is to understand the mechanism of life, like which gene, which regulatory function are needed to sustain an organism. The other way you can go into the industrial direction, like if you want to produce some medicines, like some biological compound, you can also engineer cells so that they can produce those compounds for you. So you can either go in the science or in the industry direction. So you might be familiar with something similar. You might be familiar with genetic engineering. So the idea of genetic engineering is like you might induce some mutation like on the bacteria or the virus in the lab, and you want to measure their phenotype, like maybe their resistance to antibiotic drugs, and you want to try to mutate so that you can find a new strength that can be resistant to the drug, and you want to understand which gene or which mutation that occur inside that makes it resistant. So genetic engineering is usually only about modifying just one part of an intact system. Like you want to mutate maybe some genes, but you are not looking at the organisms as a whole system. In contrast to synthetic biology, the goal of synthetic biology when it became a new field is about designing and controlling the whole system. So you want to know what goes on inside the cell, not just modifying one pathway or one function. So you need some tools to be able to do that. So in synthetic biology, they organize how they view the cells or the organisms in terms of hierarchy. At the bottom is the DNA sequence, we can design DNA sequence. At the second layer, we view DNA sequence that have different functions as different parts or Lego pieces of the system. You can have some DNA sequence that act as a promoter, some DNA sequence that act as a ribosome binding site for translation, some DNA that stop the translation, some DNA that code for genes. So we can view these as components of the system. Then what people in synthetic biology call device, you can think of as a gene, for example. A gene consists of a promoter, consists of a coding region, consists of a termination. So you have to have promoter, like start site, codons, and then the stop codon, and maybe even the UTR, untranslated region. You have to have all these parts connected together on a linear DNA for you to have a functional gene. And when you have multiple genes, these genes can regulate each other. So beyond device, we can view the connectivities of genes networks as a circuit. So basically, in synthetic biology, we're going to view the cells much like an electronics or a device. We have parts like resistor, we have diodes that we can put on the circuit board. So we're going to view the cells the same way. We have DNA, we have protein, we have enzymes that can be put inside the cells, and these will act like parts. But instead of being an electronics part, now these are biological parts. And we can view how we design the cells as a programming. We think of cell as computing something. It's going to take the code, which are in the form of DNA, is going to get some input, like which may be some substrates, that's going to produce some output, which is a different metabolite, for example. So the goal is to like design the cell so that we put in the right gene, we put in the right regulation, so that the cells can process one substrate and turn it into a different one that we want. And given a system, like when we look at gene regulatory networks, usually in biology we look at these kind of diagrams, right, and we understand that okay, if I have the substrate, then it will react with this transmembrane protein, which may be transported inside the cells, and this protein might be activating a certain other protein, which then activate the transcription of a different gene. This gene gets translated into protein. It might bind to a different location on the genome, and it activates even more genes. And in the view of synthetic biology, this type of diagram can also be rewritten as a logic diagram. So, for example, like this half circle symbols means you have to have both conditions in order to activate the next gene. So basically, you have to have a medium concentration level of the substrate and also the transmembrane protein MOR9-1 together. These will interact and then activate CREB1 gene. And CREB1 gene has to kind of float inside the nucleus, identify the DNA sequence for VAN-A2, and when CREB1 can bind to the promoter, like it can activate the transcription of VAN-A2. Like, so this diagram kind of basically describes what we usually learn as a gene regulatory network or signaling cascade, but we are now writing it in terms of electronics design. So in synthetic biology, the idea is like we are going to come up with some design, like because we want the cells to maybe survive in certain conditions or produce some metabolite or produce some compound that we want. So we're going to work on a drawing board on our computers to decide what genes, what regulation we want to put inside the cell. And then we're going to have to build that system. Like we're going to synthesize the DNA and put it inside the cells. And during the test phase, we're going to grow the cells and look at the phenotype. Like do they grow in the condition that we want? Do they produce the high concentration of the medicines that we want? And if it fails, like we can learn from the mistake. Like this design doesn't work. Like maybe we should change the enzyme. Like maybe we should change the regulation. So in synthetic biology, people usually define this kind of design, build, test, and learn cycle. So you design something, build it, test it in the lab, and then learn from it whether it succeeds or fails. Right? And then we try to change the system to optimize it further. So this will be where computational tools that we have seen throughout this course, like, come into play. We can predict. If we can design better, like using computational tools, we can save the resource and save time when we build and test the cells that we create. So let's look at these components kind of one by one. Right? So first, let's start with biological parts. Like we want to see if we want to build a new cell, like a new artificial life, what kind of tools do we have? So in the community around the world, people come together, and when they design any new genes or design any new regulatory element, they put together this kind of a catalog or registry of standard biological parts. So these are optimized sequence or genes or regulatory networks that you can order like from different labs. So these, if you go to the registry of standard biological parts website, this is a website, they will have for you this kind of catalog with the ID for the parts and some description. This is a arabinose inducible regulatory promoter. So it's a promoter that you can just copy the DNA sequence and then you can use. And it's connected to the arabinose gene. And these are the creators. So this is the lab that design these parts. And if you contact them, like they can produce and maybe send to you the pre-built DNA sequence. So you don't have to come up with all the parts yourself. Like if you want some function that people have already decided for, you can get the design and you can follow it or you can order from them. So this kind of help everyone around the world when they want to build a new synthetic cells. Instead of working from scratch, they can build on top of other people's design. So similar to how you can buy electronic parts that other people already built, and then you can connect them into your own circuit. We can now do that with biological systems as well. So for example, these are standard parts that you can find on the catalog. You can find a well-defined promoter. If you want to activate it with a certain transcription factor, there are promoters that are designed for that. If you want genes that do some function, you can find genes with that annotation. If you want to use specific primer so that you can amplify your gene, people already design primer binding site for you so that you can insert into your DNA sequence design. So this allows you to take all these parts to put together to create genes. You connect promoter to coding sequence to untransfected region, and you get a working gene, a working device that codes for some function. Then you can build the system by putting the genes together to form the pathway. Another component that you need, once you build all these parts, you have the genes that you want, you have the regulatory element that you want, you need to put it in some cell or some system. So people already also built some kind of chassis that you can put your DNA parts inside. Usually these are bacteria because they are simple, they don't have that many genes, and you can grow them quickly in the lab. So E. coli and Bacillus are popular bacteria that people use. But if you want more complex system, you can use the popular Cerevisiae, or several labs also create a plant model or mammalian system that you can put in your gene, and then you can observe how it works, how it changes the phenotype, for example. But also there are some people that work with a cell-free system. So basically it's a in solution. So there's no cell, no membrane, no nothing. But you put in the necessary ingredient, like the ribosomes, amino acid, individual amino acids, transfer RNA and ATP, some energy. And then you can induce the production of proteins and the interaction of proteins without having to have intact cells. So these are the options of where you can experiment with the DNA of the gene that you design. So other people also try to create a genome that you can kind of freely edit. So in this case, the J. Craig Venter Institute, they start with the bacteria called Mycoplasma, and this bacteria start with a million base in the genome. But they try to reduce all the non-essential genes, so that you have the minimal genome that you need to sustain the organisms. They can narrow the DNA down to half. They can remove half of the genes, and the remaining about 500 genes, this is enough to maintain this organism. So this can serve as a template that you can add custom-made genes into it, so that you can observe the phenotype that you want without interference from other genes. But even in this kind of small system, there are still about 150 genes that the authors reported that there's no known function. They don't know why it has to be inside this organism, but they cannot remove it. They need it to maintain this cell. So this is kind of some attempt to engineer a chassis that you can perform synthetic biology experiment without minimal interference from unknown genes. So for the second layer, once we are able to create all the parts from the catalog, we have the chassis that we can put the parts, the genes inside to observe the phenotype. Next, we want to know how to design the circuit. We looked at this example before that if we want to create a complex gene regulatory system to have the behavior that we want, we have to be able to design how the substrate and the protein react together, how they, when they react, like how can we make them activate another protein or another gene. So people have to come up with designs for what they call a genetic logic gate. So similar to in electronic circuits or in computer programs, like you have more than one conditions, and when these conditions are met, like, it might activate the different part of the code, like, or different part of the circuit. But we need to do this in terms of biology. So for example, like, if you want to activate a certain gene, like, with a certain compound, like, there are two possibilities. Like, do you want this gene to be expressed all the time, like, or do you want this gene to be silent until you activate? So in this design of a, they call it a buff gate. So this is, you want to increase like the signal for this gene that are tagged with GFP, like, so you can monitor the green fluorescent signal. And they want to control it with this compound, p-chromatic acid. So they pick this compound. So this compound can bind to this K127Y gene. And this K127Y, in the usual condition without the chromatic acid, is going to replace the promoter of this GFP. Like, so without the chromatic acid, this system will not produce any GFP because this system will produce K127Y, and this shut off GFP. But when you introduce the chromatic acid, like this replace the K127Y, and so this promoter can now be activated. So this is a simple design. Like you control the activation of a gene with a chemical compound. It's easier than controlling with a gene, because if you want to control this EGFP with a gene, like you have to first transcribe the gene, it has to be translated and then bind to the promoter. But if you use a chemical compound, like you can introduce it and it can work right away. So it has a fast activation time. Let's look at more example. If you want to have two conditions, like this gene, the green fluorescent protein here, you want to activate it only when you have both chromatic acid, and the nitrogen, like two different compounds basically. So you want to have the conditions where if you want to activate it, you need both. Not just one is not enough, you need both. Like, so a simple design, like maybe you have two upstream genes, both of them replace the promoter of the green fluorescence protein. So when you introduce chromatic acid, it turns off this K127Y. So, but still you still have FDER like gene that still replace it. So the only way that you can activate this protein is that you have to shut off both K127Y and FDER. And that's only happen if you introduce both compounds. So this is a simple design that you can control the gene activation with two conditions, two compounds. Sometimes you want a NOT gate instead. So a NOT gate means that if there is chromatic acid, instead of producing the GFP, if there is chromatic acid, you want to stop GFP. So it's a NOT gate, it's a negate. So if you have it, you will not produce it. If you don't have chromatic acid, this will be produced. It's an inverse relationship. So in this case, you have to have more parts into your circuit. Instead of having K127Y directly replace the GFP protein, the K127Y is going to replace this non-coding DNA that in turn can bind to the GFP messenger RNA and stop it from being translated. So these are just examples that can show you that with you can apply any conditions using any number of compounds to create a genetic circuit that you can control with the concentration of your compound of trust. So other more advanced design, like some of them you already saw in the dynamics lecture, like this toggle switch design, where you have two proteins that replace each other. So replacer 2 can replace promoter 2 of the replacer 1, and replacer 1 can bind to the promoter 1 and downregulate replacer 2. So these are switches. If you turn on replacer 2, it will turn off replacer 1. If you turn on replacer 1, it will turn off replacer 2. So it's a switch. You can make cells to enter one of the two steps. Very simple. Another example that I want to show this time is what people call an oscillator. So in this case, instead of having two genes replace each other, now we have three genes replacing each other. And cool things happen when you go from 2 to 3. So when you have two genes, when one gene is high, it will downregulate the other genes. So you have a system where one gene is high, the other gene is low. But when you have three genes replacing each other, let's imagine. In this case, we have a RACI gene, TET-R gene, and lambda CI gene. If you have high RACI, you start off with high expression level of RACI. High expression level of RACI is going to turn off TET-R. It's replacing TET-R. But when you have low expression of TET-R, this means that lambda CI can be expressed because TET-R used to downregulate lambda CI. But without TET-R, lambda CI is going to be expressed. But when lambda CI expressed, it's going to turn off RACI. So now you have an interesting condition where you start with high RACI, but you end up with low RACI. But when you have low RACI, if you continue this logic further, when the cells have low RACI, it's going to have high TET-R. And when it have high TET-R, it's going to replace lambda again. And when you replace lambda, you're going to have more RACI. So you can imagine that somehow you're not going to have any stable situation. You're going to have low and high, low and high, low and high, going forever. So this is an interesting design, which is why they call an oscillator. Because if you let me try to click it. Let me try to activate the video somehow. Okay, now I just realized that I cannot activate the video with the laser pointer. This is interesting. Okay, now I can. So basically, this oscillator design, like we saw earlier, that we see that if one gene is high, the gene will be low, but the third gene will be high, and then it will make the first gene low again. So in this experiment, they show that if you let this system run, and you monitor the expression level of one of the genes, it's going to be oscillating between low and high forever. So this kind of design allows you to control the timing, like the rhythm of the cells. If you don't want one protein to be activated all the time, like you want it to be on and off, this type of three genes replacing each other can achieve that. So this is a cool kind of circuit design that is possible. And just to recap, like from the lecture last a while back, like we learned about differential equation. So if you want to understand the dynamics of this circuit, where three genes replace each other in cycle, we can write the change in gene or protein expression over time using a similar equation that we did last time. So this will allow us to identify, like based on the binding strength of these genes, we can control the period, like the time period between each cycle, and we can control the expression level, the maximum expression level of each cycle. So you can use this kind of a differential equation to analyze like the behavior of the system and maybe change the gene, change the promoter, so that you can achieve the right period and the right expression level. So what we learned kind of last time was that when we write this kind of differential equation, like we can monitor the behavior of the system. But in real life, right, the genes are not going to follow exactly this behavior. Right? For example, let's think about the promoter. These replication rely on the protein binding to the promoter and then replace the gene. So it depends on the dynamics for the binding for the protein with the promoter. If the binding between the protein and the promoter is very fast, the promoter can be on and off, you're going to have some variability, but the behavior of the system is going to behave like the differential equation that we wrote. The blue line here is a theoretical behavior that we can predict from differential equation. But the orange line, this is going to be the actual behavior of the cell. Because not all promoters are going to be bound, and depending on how long the protein binds to the promoter can stay in the on or off state for different time period. So you can have some period with high expression, some period with low expression, some period with high expression. So there's going to be some variability, some stochasticity in the real system. But if you design the promoter and the protein poorly, these two sometimes bind, sometimes don't, you can have an extended period where the promoter is on and you have a lot of production, and you have a long period where the promoter is off, and then you have a low expression level. So you can end up with this weird situation where when the promoter is on, you have a lot of protein produced. When the promoter is off, you have very low number of protein produced. So instead of behaving according to the expectation, the system now behaves in two modes, with high expression or low expression. So this kind of stochasticity, when you design the circuit, you have to be careful about this kind of behavior. Okay. When you want to kind of design the circuit and when you want to see the behavior, fortunately there are these kind of tools like BioNetGen, Theorem, and Copasi. So these are bioinformatics tools that allow you to draw the circuit. You can take the genes, connect them to the promoter, and connect to the next gene, and then you can define the binding strength. And then this will simulate the differential equation that are based on this circuit for you. So you can tweak, you can change the genes, you can change the binding strength, and you can identify which parameter gives you the best kind of behavior that you want. Okay. So this is the second aspect. We learned about the parts that we can use to build a biological system. We learned about how we can think about biological system as a circuit that we can optimize with these genes. So next, we want to look at the endpoint, the other endpoint of the system. We want it to produce something. So maybe we want to analyze the cells from the perspective that does it produce the metabolite that we want, and what is the efficiency, what is the substrate that the cells need in order to produce the metabolite that we want. So when people talk about metabolic engineering, they are viewing the cells as a factory. We are going to use the cells to produce some metabolites as a product. So we have some substrate that we feed to the cell, and we have enzymes and genes that we put inside the cell because we want it to digest the substrate to create the product that we want. So what we are interested in here is when we put multiple enzymes into the system, and we have some knowledge about the enzyme, so we hope that it will digest the substrate and then create the product that we want. There are two things that we want to understand. First, what's the dynamics of the system? If I put in 10 molecules of substrate, how many molecules of product would I get? We want to identify the equilibrium. If we let these cells reach for a long time, what would be the level of product that we are producing at the steady state, at the equilibrium? So when you see the term metabolic flux modeling, so flux means changes over time. So basically they are going to try to understand the dynamics of the cells if we put in some substrate. And compare to when we learn about gene expression, we have some differential equation model, and we assume that we know something about the binding constant, like we know something about how genes activate or replace each other. But in terms of metabolic pathway, right, sometimes we don't really know what's the efficiency of the pathway. Okay. Sometimes we don't really know about the dynamics of the pathway. Right? If we put in 100% of the substrate, right, how much would be turned from A to B? Right? How much would be maybe transferred to a different pathway? Right? How much would come out as the product H at the end? So we want some experiment, right, that we can perform so that we can understand how the substrate flow through the metabolic network. So in this case, right, people use isotope labeling. Right? So if you label the substrate with the isotope and you feed it to the cells, right? Similar to when we use this to analyze mass spectrometry data. Like we treat the cell with different food, like with heavy and light isotopes, and then we can study the protein with different isotopes. So in this case, like when we put in the substrate with isotope label, like when this substrate got used, like got processed inside the cell, like some part of it will transfer to kind of different part, like inside this network. Like some go from A to B to D, and D to E. Right? Some may go from A to B, B to D, but D go to F, right? And F go to G. So in this case, because we label the original isotope, original substrate with the isotope, we can monitor how much metabolite, right, in terms of A, B, C, D, E, F, G, right, how much percentage is labeled. So we can monitor how fast the substrate got used, right, and what fraction got turned into different metabolites. So we can monitor the fraction of the isotopes. Okay. And once we have this information, right, you can now model that if I put in 100 substrate, 80% is going to go from A to B, but 20% is going to go sideways, like from A to something else. From B, like from 80 compound, B can produce more product, like from 80 maybe to 140. But from 140, maybe 70 goes to E, and then 30 go to H, for example. So by following the isotope label, you can model, you can reconstruct the metabolic pathway and know how much metabolite or substrate can flow into each branch of the pathway. So once we have this estimated reaction of the metabolite, we can now try to understand what would be the equilibrium of the cells. The equilibrium in this case means that the cells have a stable level of each metabolite, no excess, no. So we can write the differential equation again, because now we know the rate of the substrate, 100, and we know the how much metabolite will be produced from A to B, from B to C by following the isotopes. Now we have these numbers of the reaction. So we can write this equation where what would happen, what would be the level when A doesn't change. So dA by dt, this slope should be 0. And this slope is 0 only when the rate of the in is equal to the rate of the production. F1 and F2 are equal. And when B doesn't change, that means that the rate of production of B, which is F2, equal to F3 plus F4 and then minus F5. So we can write this equation. So we know that if we want the cells at equilibrium, if we want it to produce C and E at certain level, this F objective, this is the amount of production of C and E that we want. We can solve this equation so that you can select genes or select enzyme. So that F1, F2, F3, F4, F5, all the way to F8, these together and this equation satisfy the objective that you want. If you want to produce C and E, let's say 2 molecules per second, you can optimize the reaction by using the enzyme or changing the gene regulation networks so that you can achieve this balance with the target of 2 molecules per second, for example. So this is what people mean when they want to optimize the enzymes or the reaction of the cells. They have some target production that they want, and we want cells to produce this much of output at equilibrium. So we can solve the differential equation to get the amount of flux or amount of flow of this metabolite that can achieve the objective. Okay. So just to show you some example, you don't need to do this by hand. There are bioinformatic tools that if you put in the metabolic pathway and you put in the flow rate that you work up from experiment, they can solve for you what would be the equivalent state for the cells. So these colors represent different levels of reaction. So blue is a low reactions, orange is intermediate, and red is high reaction rates. So you can understand the dynamic of the cells, and you can visualize it using these tools. And you might have like one question with the isotopes, like you can work out the level of different metabolite and substrate. But how do you know that this is the metabolic pathway inside the cell? So people try to create also a database that collect all the enzymes that and what metabolite got processed and produced by the enzymes from different organisms. So if you go to database like Recon 3D, you can build a metabolic pathway inside a certain organism by looking at which enzymes exist inside that organism and which metabolites are produced and used by that enzyme. So you can build your own metabolic pathway from this knowledge and then put it with the experimental data so that you can reproduce the reconstruct the reaction rate for your metabolic pathway. So this is a way that people use to study the cells to make it produce the metabolite that they want. Okay. And we can also integrate like the knowledge of omics and bioinformatics inside. So usually when people analyze metabolic flux or metabolic change, they can work on metabolic only. If they measure all the metabolite, they understand the dynamics of the system. But when they want to control the system, if they want to increase the production of certain compound, for example, if you work on the level of enzymes, you might need to change the enzymes. Maybe you want faster production. You need to identify a new enzyme that can produce this substrate faster. But instead of doing that, you can also use bioinformatics to help, because if you want more activity for the enzyme, instead of changing the enzyme, you might want to upregulate the enzyme. And to upregulate the enzymes, you need to know gene and protein expression network. So people who do metabolic analysis sometimes rely on the knowledge from gene and protein layer to control the expression level for the enzymes itself, so that they can control the activities for the reaction. And with bioinformatics data, you can also model the metabolic flux or metabolic pathway in a cell type specific or condition specific manner, because different cells is going to have different level of gene expression, and different level of gene expression can affect the level of enzymes that are being produced. So for example, there are studies called optimal knock and fast knock. And so from the name, you can probably guess. So these studies want to identify if you want to change the metabolic balance inside a cell, which gene should you modify to achieve that effect. So in this case, they want to rely on gene knockout because it's easy to perform. So inside a bacteria, if you want to increase maybe the production of ATP, which gene should be knocked out, so that the cells produce more ATP. So these are bioinformatics tools that makes prediction. If you want to increase or decrease a certain metabolite, which gene should be knocked out from the system. So this is an example of how bioinformatics and computational model can help experimental people. Instead of having to try different enzymes and see which one works, they can make predictions and modify their synthetic biology system and test if that achieves the effect that they want. So for the last section, we're going to talk about the roles of computation in synthetic biology. We already saw some examples. How can we model the dynamics using differential equation, like how we can predict the genes to knock out to achieve the right production of the metabolic. But we can do more with modern tools. Like we can design the genome. So the idea of designing the genome is you want to create genes or create regulatory elements that have certain property. For a gene, like you might want it to encode a certain protein with certain function. With genes, you might want to have a certain messenger RNA secondary structure or stability, which in turn affect the expression level. For regulatory element, you might want it to have a binding site for certain protein, or you want it to be complementary to certain non-coding RNA, so that you can use those non-coding RNA or those proteins to regulate your gene. So this requires some knowledge. You have two options. You can either use existing DNA sequence, like maybe you found a suitable gene or suitable regulatory element from some bacteria or from some plants. You can extract, you can clone those sequences, and you can use it in your synthetic biology experiment. But from another angle, like you can do a computational search. Like you have a working enzyme, but you want it to have more or less kind of reactivity. You might want to try to mutate that gene or that enzyme and see if you can reduce or increase its activity. So this is where computational tools can come in. Imagine if you start with maybe a random DNA, or maybe you start with a known gene, and you want to make prediction of which part of the gene should be mutated to create a new gene that have the property that you want. So if it's a protein function, when you make the mutation, instead of having to test the function of the protein in the lab, you can use AI to predict maybe the protein 3D structure and predict the protein function. So you can try a lot of mutation and predict the ones that and find the one that have the predicted activity, and you can test only those proteins in there. So it can help narrow down the pool of possible mutation. Another way, if you want to insert a regulatory element inside your gene, you might have a known protein binding site, a transcription factor binding site, that you want to add to your gene so that it can regulate your gene. But you have an option, where in your sequence should you add this binding site. Instead of trying several regions, you can do prediction. You can add this transcription binding motif, and you can make prediction whether this can be bound effectively to the protein, and what will be the expected expression level from this transcription factor binding site. These are something that we can try to predict. So you can pick the best transcription factor binding motif and put it in the best location on your gene to achieve the regulation effect. Or you might want to design new sequence from scratch. Nowadays, there are many studies that try to generate a novel DNA sequence that have the property that you want. Maybe it has a binding site for certain transcription factor, and it will achieve certain expression level. But instead of using known genes or known sequence to modify, these tools can create something new from scratch. So let me give you one example. Let's say you want to optimize the untranslated region. So untranslated region, even though it doesn't code for the protein, it can affect the stability for the RNA. It allows the RNA to stay longer. And staying longer means it gets more chance to be translated. It also can form secondary structure that recruit the ribosome. So if you optimize the untranslated region well, you can tune your gene expression level without changing the gene body itself. You just change the UTR. So there are some people who try to create a new AI model that can predict, can generate new UTR. And that UTR you can control if you want to have high expression, like high ribosome binding, like high translation efficiency. And people train this, for example, if you use gene expression data from human cells, because each gene has different UTR. So we can already train the model that if they look at the UTR, can they predict the expression level for the gene? So this kind of model will understand some aspect of the UTR DNA sequence that affect the gene expression. So if you train this model and you ask it to produce, okay, I want a new UTR that have certain level, that will produce a certain level of gene expression. It's going to be able to mimic known UTR that are associated with known genes that have a similar expression level, but it might introduce some new mutation inside. So people are using this to, I think we mentioned this in the RNA lecture. People use this type of tools to optimize the mRNA vaccine. When we want to inject the vaccine in our body, we want the vaccine to be stable inside our cells, and we want the messenger RNA to be translated by our cells. So we want to optimize the UTR so that we can achieve that. So this kind of ability to design new molecules, like new DNA sequence, can help speed up the design, build, test, learn cycle in synthetic biology. So instead of designing with the knowledge only, you can also design using computational prediction to drive your design, to pick the one that you should test. And there is a concept called active learning. So you might have heard about this in the context of education, where active learning usually means that both the teacher and the student have to take part in the learning process. So active learning in computational as a computational technique is when, if you have an AI, it might perform well on some genes, it might perform poorly on unknown genes, on unseen genes or unseen organisms. The idea of active learning is, now that we have synthetic biology system where you can put in new genes and you can observe the phenotype in your system, how about we also try to predict which experiment should be performed, like which gene should be tried, which gene that we should put inside the cell, so that the knowledge that we get, like the phenotype that we get, will help improve the AI further. So not only are we trying to find genes or biological parts that achieve the property that we want. In this case, we want to improve the AI by saying that, okay, I have synthetic biology system. I want to improve the AI. So I want AI to be able to predict better on some class of genes or maybe on some organism. So I can design the experiment so that the data that I'm going to produce is going to be also beneficial for training the AI, for improving the AI. So for example, when we have an AI model that maybe can design chemical compound for drug discovery, we know that the universe of all compounds is not uniform. There are certain chemical structures that people explore a lot, and there are certain chemical structures that not many people study. So if you imagine this as a space of chemical compound, there might be certain regions that are not explored. These drugs are not well studied, so no one really learn about them. So this can affect the AI, because AI will be fitting to other compounds that people study a lot, and the AI will be able to make prediction very well for those compounds. But if you pick these understudy compounds in your research, you add them to your synthetic biology workflow, you can produce new data that are beneficial for improving the AI further. So when you have kind of this integration between prediction from AI and experiment to confirm the prediction. You can do it intelligently to make sure that the validation that you perform also benefit back to the AI model. Okay. And another situation that AI can help a lot with synthetic biology is in the lab in terms of automated experiment. So nowadays, many labs around the world is exploring the idea that if we have an AI that control the robot arms, we already have this in many labs. You can have the AI to pipette or put the cells or the sample in the right position, so that we human can do the next experiment. But how about if we let the AI also put in the substrate, put in the compound, put in the cells with different conditions. So we train the model to control the robot arms, not only to transfer the sample, but also to put in all the substrate, all the ingredients, all the compounds that are needed to test the system. And if the phenotype are easy to see, for green fluorescent protein, if the robot arms can take the plate onto the microscope and take the image, you can imagine that the AI now also see the phenotype of the cells. So people are now developing models that let the AI perform the assay, perform the molecular assay, look at the result, and use that result as a feedback on its design. If it put in certain concentration of the compound, and now it sees that the fluorescent signal is low. This is a feedback that it can increase the concentration of the compound. So people are designing ways to automate this type of experiment, where if you want certain phenotype and you don't know which compound to use, instead of having to define which compound and which concentration to use, we let the model control the robot arms, take in some concentration of the compound, look at the assay result, and then update its concentration and compound choice automatically without human supervision. We can have an AI that can discover new drugs or new effects on its own. So this is another way that computational thinking and modern AI techniques can improve the throughput and the efficiency of synthetic biology. Okay, so this is everything that I want to talk for today. So synthetic biology is about designing, manipulating the whole system, not just one gene, not just one pathway, but the goal is to understand the working of the whole system. So we can do this in the engineering way. If you want to understand a certain system, the best way to understand it is to try to mimic it, to design some new system that have similar properties, and try to take some part out and try to take some part in, and try to understand which one affect the phenotype that comes out. We saw the concept of biological parts, we can view promoter, genes, regulatory elements as parts that we can assemble like a circuit. And so you can view the cell like a computer, like a electronics board. You can put in genes, put in components, and put in the regulation, put in the rules, put in the connectivity, and you can create a whole cell or a whole organisms that behave in the way that you expect. We saw the concept of metabolic engineering and flux optimization, where we want the cells to behave as a factory to produce the metabolite, the compound that we want. And we want to understand the efficiency. We want to optimize the enzyme. We want to optimize the metabolic pathway. And we want to optimize the flow rate of the substrate and the metabolite, so that the cell as a factory does what we want at the rate that we want. And we saw some example that knowledge from omics can help metabolic engineering, because now we can control the expression level of the enzymes, through knowledge from gene regulation, from knowledge from gene expression. And we saw the use of mathematical modeling, differential expression, that we saw as when we use to study gene expression dynamics. Now we can also use it to study the flow and the balance of metabolites inside the cell as well. and the benefit from AI or modern prediction tool that can accelerate synthetic biology experiment. Okay. So that's everything that I